Hi, morning, great talk. Just want to ask one questions about your CCREs. Among the various CCREs, you mentioned CTCF, and you've shown in the plot, like about 30 slides ago, that they overlap an insulatory area behind the enhancer, right? I just want to ask, CTCF has been recently associated also with TADs and chromatin looping.
Yes.
Right? You also show in one of your brilliant slides the HIC experiment. So are those insulatory regions the same as the chromatin looping closures? Or if not, have you perhaps looked into some associations between the two? Thank you.
The chromatin looping sites are presumably a subset of what we show. So our CTCF bound sites encompass the looping sites. Yes, they're the same factor. It's like a factor that can do many things.
All right, thanks.
Thank you.
Hi, I have kind of general questions because, like, before the ENCODE projects, most of the people think that only a small percentage of the human genome actually serve biological functions.
Correct.
But after so many consortiums studying the annotating the human genome, as of now, do you know how many percent of the human genome actually serving biological functions?
Yeah, it's a very difficult question to answer. So our CCREs roughly cover 20% of the genome, but now if you have a 300 base pair or 200 base pair region, maybe some of it inside is like padding positions. Not all the positions are precisely functional. It depends on the position of your definition.
It also depends on the assay. I mean,
correct. It depends on the assay.
Yeah.
Like Paul was asking, binding, is binding good enough?
Yeah.
Does it have to do something, right? So that's why we, ENCODE has been kind of criticized in the past for claiming too much territory of the human genome to be functional.
Right.
So that's why one of the motivation of our work is to combine these kind of biophysical, biochemical assays with evolutionary conservation. And evolutionary conservation is the other camp of people. Like if it's not functional, then it's not conserved, kind of. So we aim to combine the two approaches to truly define the regions that are likely to impact human traits.
So a follow-up question is, in the future, what ENCODE is working on? What are the assays that you will add to the annotation? And
Yes.
for example, cell type,
Right.
developmental time, or 3D genomes. Are these things in
Yeah. So ENCODE is done. ENCODE finished. ENCODE 4 is done. So ENCODE concluded itself, declared victory in February of 2022. So we're done. There are additional consortia to carry on this kind of work. So there is one called IGVF. I'm also part of IGVF. I'm a consortium animal. So if you want to ask about assays, so a lot of new assays are like functional assays, CRISPR and reporter assays. So they try to link directly a region with a reporter, or if you knock it out, you're going to see a decrease in function. So more related functional assays, and then also single cell. Because a lot of the times right now is like a tissue, cell type, it's not detailed enough, not enough precision. So there are a lot of single cell assays that are being carried out by newer consortium activities.
Thank you.
Yes. Yeah, non-coding RNAs are part of the data. The data is so rich. If you're interested in non-coding RNA, go in and yeah. We a lot of the times we're like 北京呀, 30. I don't know if that makes sense to you. Like the same data set, we will look at it from every each way, and every time we get a new conclusion. It's really a bolus of very, very rich and high quality data. We continue to annotate annotate the data. I mean, I have a grant from NHGRI to continue the work we're doing in ENCODE, trying to make it more useful to the community. But I can attest to a lot of the data sets of being really high quality.
So the transposable elements,
Yes.
I have a template sequence or kind of a consensus sequence.
Yes.
Can that accommodate like any transcription factor binding site, or does it affect the overall distribution of suitable bindings that for each, if you look at all the transcription factors, how many candidate binding sites do they have? As the expansion, yeah, you get it.
Yes, yes. So there is a preference. Some TEs they prefer to carry a subset of transcription factor binding sites. So there is a kind of preference match between TE families and TFs, and 1,600 TFs, and then all these TE families, and even between human and mouse, like sign elements. ALU is specific to human, and B1, B2 specific to mouse. I mean, it's going on a lot. It's a kind of preference for sure. I'm very impressed by the capacity of TEs to carry different types of transcription factor binding sites. Most of the TFs have a portion of their elements being carried around, and obviously, if we look across the TFs, there's a big range as to the percentage of their sites being moved around. Yeah, so somehow TEs make it work.
Thank you.
So let's thank